Okay, I'm Stover Zello.
I'm Valerie Wendell. Hi.
Valerie. We had some video issues before, so we fixed things. So we're going to start at number 2 while we rewrite number 1. And I'm just going to go through that with you here. So for number 2, third time round. You guys don't know this, but it's the third time for us. A is sorry. B is MIC2. C is a cross between the IGF2 heterozygous male with a heterozygous female. And what are the phenotypes of the offspring? We have that right here is 50% normal with 50% dwarf. This is the cross here. The males are the ones that carry it. The female basically doesn't matter on that one, just for that gene. So D, we got sex influenced. E, we got genomic imprinting and dosage compensation. F, we got the exist and the T6. And G, we got autosomal chromosomes and X chromosomes.
Okay, so then we're back to 1, and let's see, it is, or 1 to 3, oh, 1, okay.
We can do 1.
Okay, so on 1, it is a question about Angelman syndrome, and the Angelman syndrome is recessive. However, it's represented by the little a, and it silences the paternal alleles, which means all the alleles that come from the dad, it silences. And so for A, it says the following cross is done, big A, little a, and then what are the genotypes and phenotypes? We have the genotypes here. The phenotypes are 50% Angelman, 50% normal, because, like I said, the paternal alleles are silenced. For B, this question is concerning part A as well. The following cross is done. For the male, it's big A, and the female is little a. And then what are the genotypes and phenotypes? We have the genotypes here. The phenotype is 100% Angelman syndrome because, again, the paternal alleles are silenced and maternal alleles are all Angelman syndrome. And for C, suppose a woman has XXX syndrome or 3X chromosomes. However, the X6 have been deleted from 2 of the X chromosomes in each of her cells. How many bar bodies are found in each of these women's cells? We've got 1 bar. And then D, suppose you isolate a cell from a female cat who has 2 X chromosomes. However, the T6 genes on each X chromosome are non-functional, while the exist genes on each X chromosome are functional. How many bar bodies are expected to be found on the cell? And we got 1 bar. And so now we're on to 3. Oh, I can do 3. All right. Okay. Okay. So then 3, it has to do with the OTD that the woman's father has. That's recessive, X-linked recessive, and so if the father has it and the mother is homozygous and is unaffected, then these will be the genotypes. We have little t for the affected and big T for the unaffected. Then B, if a woman has children with a man who does not have OTD, what is the chance that their first child will be a son with OTD? And so we have that there will be a 25% chance that their first child will be a son and that he will be affected. And then C, identify the genotype of the male with whom the woman could produce a daughter with OTD. So the male would have to have the affected genotype on his X cell, or X chromosome. And then D, consider the cross described in part C of this question. What are the expected genotypes and phenotypes of the offspring? So we have the genotypes here that we've written out, and then we have 1 female OTD, 1 male will have OTD, 1 female will be normal, and 1 male will be normal.
All right, so number 4, Riley's going to go ahead and write number 5 since we don't have enough space on the board. But number 4, this one's talking about the presence of beard in some goat species. Is determined autosomal gene that is dominant in males and recessive in females. So for the first one up here, we're looking at, suppose that you have a cross that produces 3 beard, 1 no beard phenotype ratio among male offspring. So we have the male in black for offspring, and the later one's female is red offspring. What are the genotypes and phenotypes of the parents? So genotypes are listed here for female and for male here, and then the phenotypes, bearded male and non-bearded female. For part B, suppose that a cross is done, produces 1 beard female and 3 no beard female. What are the genotypes and phenotypes of the parents? So genotypes right here for female, and for male right here we have big B, little b for each. And the phenotypes is no beard on the female and beard on the male. For C, we have a cross between a male with a beard and a female without a beard produces one beard, one no beard phenotype ratio among the female offspring. What are the genotypes of the parents? So that one we have here, these are all considered female offspring. We have genotype for the woman is no beard, and genotype for the male is big B, bearded. Well, I guess it's just genotypes. So just the genotypes are big B, little b, female, big B, male. And for D, we have suppose a male and female both, which lack beards, produce one beard, one no beard ratio among the male offspring. Where the genotypes, just the genotypes of the parents, and we got for the male little b, and for the female we got big B, little b. Okay, so almost done here. Perfect. Okay, so Valerie's going to do A and B, and then I'll do C and D.
Okay, and so this A and B is regarding the cats, and it is, the color is X-linked, and so if it's a big B, it's black. If it's a little b, it's orange. And then for females, if they have both big B and little b, then it's a case called tortoiseshell. Tortoiseshell. So we have a cross between a tortoiseshell female and then an orange cat or orange male, and then what would be our outcome genotypes and phenotypes. So here's our genotypes. Our phenotype is that we have one tortoiseshell female, one orange female, one black male, and one orange male. And then for B, it asked him, how is it possible? It's very rare, but some male cats do have tortoiseshells, and how is that possible? And we figured he'd have to have two X's and one Y, and one X would be big B, one X would be little b. It's a rare case called Klinefelter syndrome.
Okay, so 5C, in the fruit fly, the mutation produces a scallop phenotype. Assume that the scallop phenotype is the result of X-linked recessive mutation, so X-linked recessive. Indicate the genotypes and the phenotypes of the progeny produced when reciprocal crosses are done. So reciprocal cross, you have an unaffected male and a affected female. So when you cross them, the female should be, we have X little d, and the male is X unaffected big D with Y. So when you cross those, you should get the phenotype, the genotypes of the progeny are right here, the F1 generation. So we have, for the males, you have X little dy, X little dy, and the females, you have X big D, X little d. And the phenotypes of that would be normal females and affected males. And for D, same stuff concerning part C. So assume the scallop phenotype is a result of autosomal instead of X-linked. Indicate the genotypes and phenotypes of the progeny produced when the reciprocal crosses are done. So reciprocal cross, same thing, but autosomal. So you'll have X little d for the females, and you'll have X big D for the male. You cross those, the genotype's all the same. It's X big D, X little d, and the phenotype of all is normal or unaffected.
Perfect.
Okay, that's all the ones we got. So
Enjoy.
see you next time.